Okay, I think we'll go ahead and get started. Welcome, everybody. Really excited to have everyone back after our break last week, and especially excited today to welcome Dr. Amanda Lyn Gunn, who will be speaking to us about rebuilding broken barriers, how the nucleus maintains its integrity. And I'm Jeannie Chowning, Senior Director of Science Education at Fred Hutch, and just like to welcome everybody to our event today. Just a quick reminder, sometimes we have people in our audience who are not science teachers or students. This talk is geared towards science teachers and students, so please keep that in mind if you are asking questions or participating. Be conscious of the main audience here. If you're not talking, please mute yourself. This is especially true if you're connecting via a phone. Please feel free to add questions into the chat as we go along, and when we get to a good stopping place, we'll make sure that our speaker can answer those. And then a reminder to please complete our feedback form at the end of the talk. And you'll get another reminder then. All right, so as I mentioned, today's speaker is Dr. Gunn, and Dr. Gunn is a staff scientist at the Hatch Lab at Fred Hutch, and I'm super excited to hear her presentation. And I think we'll go ahead and turn the screen over to you.
Let's see, screen share, audio, everything. Okay, can you see my screen?
Yes.
Okay, perfect. So as Jeannie mentioned, my title is Rebuilding Broken Barriers, How the Nucleus Maintains Its Integrity. And I'm Amanda Lyn Gunn. I'm a staff scientist in the Hatch Lab, as mentioned, and you can all follow me on Twitter if you would like. I communicate with a lot of people a lot of times, so feel free to follow me there and interact with me at any time. Basically, if you do that, you should be warned. I post about science, I post about advocacy for diversity and inclusion, science communications, a lot of dog pictures, I will be honest, and forewarning, my dog is home and tired and cranky right now, so if you hear some noise, I apologize. I also work with a mentorship program for graduate students and postdocs called Bending Singletons, and some more personal stuff about me there. But like I said, feel free to follow me if you ever want to reach out about anything. So what I'm going to do today is I'm going to talk a little bit about my background and some of my outreach work before getting into the research I do in the Hatch Lab at Fred Hutch. And there should be time for discussion if I don't ramble on too long about anything. But one of the first and most important things I want to get through is the acknowledgments, because this is Team Hatch Lab. We have our technicians, Heather and Emma, and our graduate students, Lucian and Molly. And then we have postdocs, Allie and Anna. And of course, our fearless leader, Dr. Emily Hatch, who runs the lab. And I'm going to explain a little bit later on what a staff scientist does, but that's my role in the lab. So getting into my background, I actually, when I was in high school, I'm from, originally from the eastern area of Los Angeles. And I didn't go to the sort of school where everybody is super driven. There were, of course, the kids who were driven and born and raised to be in a certain career, but I actually didn't know what I wanted or how to get there. And so I made some of, I don't even want to say some of, I made every mistake a young person can make as they're beginning a career. So I got my bachelor's degree from the University of New Haven in Connecticut in forensic science, criminal justice, and chemistry. But that was after I transferred schools about 7 times. I lived in Hawaii, I lived in Connecticut, I lived in California. I moved all over the place because I didn't actually really understand how the system worked. And so this really shaped a lot of how I approach my career and how I approach mentorship now, because one of the most common things you can hear me say is you don't know what you don't know, and that's not your fault. So unless you have people to help guide you through whatever you're going through in life, you can get pretty lost. By the time I graduated, I could have, if I stayed all at one school, had 13 minors because I just loved to learn and I just kept taking classes. But I really loved science. And my first job after getting my bachelor's, I worked at LabCorp doing diagnostics, and then I worked on developing DNA kits, and I eventually stumbled into a PhD program at City of Hope Medical Center in Los Angeles, which is actually a very similar institution to Fred Hutch. The reason I chose City of Hope, because I didn't know what a PhD was necessarily when I started looking into it. The reason I found it was on the education page of City of Hope Medical Center, their website, and I was snooping around there because that's where my grandmother was treated for cancer when I was a kid. So that's actually how I wound up getting a PhD, even though just a few years prior to that, I didn't really know what a PhD was. I did have some internships before that. I managed to get in, who knows why, but I wound up getting my doctorate in biological sciences, focusing on molecular biology and genetics. From there, I really wanted to move forward with education, so I got a position as the laboratory director at Claremont McKenna and Scripps. Claremont McKenna, Scripps, and Pitzer colleges. And after that, I wound up going to a tenured position at Grace Harbor College before I came here, and I'll give you a little bit more about that. So first, my work in graduate school was very focused on genetics. I was in a super supportive environment within my lab. We had a really great team working on DNA repair mechanisms, and my work specifically focused on the biological context surrounding DNA double-strand break repair, which is another theme, is I'm always looking into the context of a thing rather than just the strict mechanism. This was a really great experience for me, and I was able to be super productive. And so when it came time to move on, I wanted to do something that was a little more challenging for me, which involved getting involved with education. So I took the director position at the Claremont colleges in their integrated science department, where I predominantly focused on building laboratory education for the undergraduate students that integrated biology, chemistry, and physics. Because if you remember, I took literally everything as a student. So I helped create these package units that our students could learn how to work not just in one field, but how to integrate a lot of different things and change the context of an experiment. And that was really rewarding, but I missed research. And so at a faculty dinner one time, I happened to meet Professor Larry Grill, who is one of the coolest people I have ever met. And he was talking about his research. He develops plant-based vaccines and teaches people in other countries how to develop their own vaccines instead of going through the great big system for underprivileged countries who need access to things like a vaccine for rabies or rotavirus. They're able to actually create the vaccines themselves. And so I half jokingly told him, I said, wow, I would just love to come and learn from you sometimes. And he said, okay. And so I began working in his lab and developing these vaccines through genetic cloning and growing them literally on plants. And this was really cool. However, my family decided, for a number of reasons, to uproot ourselves from Los Angeles and move to the state of Washington about 7 years ago now. Just through a random series of events, we all decided to up and move here. And, excuse me, and so I decided to find a job here in Washington as well. And that job wound up being at Grace Harbor College, where I was initially hired as just a biology professor, with the hopes that I could take all the really cool things I learned at the Claremont colleges and make them available to a 2-year institution, because that's one of the most important things. As somebody who didn't have a lot of mentorship getting through my undergraduate or high school or any of that, I actually didn't know about programs like the Hutch programs that you're participating in, or internships. I didn't know about RAU programs. I didn't know about most scholarships. And when I was at the Claremont colleges, you know, not as a student, but as an employee, I suddenly learned about all these things that I missed out on, and that my pathway could have been so much easier if I knew about it all. And I thought, why wouldn't I have known about that? You know, I was in the science programs at a lot of schools, so how could I not have heard about these things? And it occurred to me that's really one of the major gaps between going to a community college or a junior college and a 4-year school, is the focus on those extracurricular activities and providing those extra opportunities. Or at least it was at the time. A lot has changed since then. And so I went to Grace Harbor College, and I decided my goal was going to be developing opportunities for students that were similar to what the students at the Claremont colleges were getting, and have research opportunities available and job support, and really getting together this program that's sort of cohesive but open to everybody. And so I was leaving this world where everything was very cut and dry and clean and tidy and mostly in a test tube, like the plasmid on the left. And I entered this world that was centered around fish, and salmon in particular. And so in addition to that change, I went through this world on the left where I was cloning things and everything had a protocol, and I just followed the arrows, and if you need to troubleshoot it somewhere along this path, and everything again was clean and tidy, to suddenly finding myself as a tenure track faculty member where you have to make a lot of decisions. What experiment to do next? How are you going to fund everybody's salaries? How are you going to do the outreach? How are you going to design this program? It's a lot messier, and there's a lot of higher level thinking in a career as a faculty member than I ever even had expected. But I loved it. So you might be wondering why fish at this point. So what you're looking at here is a, looks like a male chum salmon, and there's a female on the upper left there. These are chum salmon in particular. We raised these eventually. When I got there at Grace Harbor College, I had these great big ideas that I knew what I was doing. Nobody ever knows what they're doing. I showed up and I thought, hey, I'm going to create this program where students can get involved in research. And I had this great model that I'd learned from Dr. Grill to create plant-based vaccines. It's inexpensive, it helps the world. I thought it was going to be great. But moving to this rural area, I very quickly learned that many people did not like vaccines. And they also very much did not like a girl from Los Angeles coming and trying to teach about vaccines. So I actually got really frustrated in the beginning. I put out all these flyers. I was trying to recruit my students. And I had 2 students show up in the very beginning, and they weren't really even that into it. They kind of wanted extra credit. And so the first thing I really learned as a faculty member is that it doesn't really matter what I want to do. It matters what the people need and what they're interested in, and how could I shape something around the people of Grace Harbor. And you don't know what you don't know. And so I spent that first year integrating myself into the community and really finding out what people cared about. And one of those things is definitely fishing. Fishing is one of the few activities, if you don't do it personally, somebody in your family does. It provides food, it's recreational. The salmon industry is very important to Grace Harbor. And so I started a fish hatchery. I had never seen a salmon before I moved to Grace Harbor, Washington, and that is one of the more amusing things in my life, because the next thing I knew, I was renovating a fish hatchery, and I started a fisheries research program. We were open to anybody who wanted to participate, and that was one of the biggest things. We called everybody a volunteer, whether they were a student, a paid intern, it didn't matter what your role was. Faculty members came and participated. We had members of the community come to participate. The grounds staff, the maintenance staff from the school, custodial, everybody came and participated, and we were all equal. We were all treated the same. And anybody could come, and I would come up with activities or research opportunities, or whether we were raising salmon or planting trees, something for everybody to do whenever they came. And we even, as I learned, once you really invest in your community, you can also kind of shift that towards things that interest you as well. We even identified several new species of prokaryotes, of archaea specifically, in our stream throughout the 5 years of research that we did. And by the end of it, there were over 2,000 participants in this program. We collaborated with Washington Department of Fish and Wildlife, Chehalis Basin Fisheries Task Force. Whenever a bridge or a culvert needed replacing, we went out there and planted the trees to restore the area. I actually retrained enough that I wound up teaching an entire fisheries series of courses. And most importantly, my students, my participants, my volunteers were all thriving. So just a few examples. Maddie and Lauren Thompson are now students. They're graduating next year at the Evergreen State University. And Lauren is actually doing a lot of science communication for them. They both have paid research internships now. Evan Bratton, I met when he was in high school, and he is now working as a research specialist on a fishing boat. Marina Latimer is finishing school. Ted Elson is working in forestry. And Morgan Melitza is about to finish her 2-year degree at Grace Harbor College. So we have all these people who came and participated, and no matter where they were headed, we made an effort to really help them get there. And we also, in that time, raised about 100,000 fish, which is pretty cool. So how did I get from all those fish and all those years of experience to where I am now in the research I'm doing now? So one of the things that in my career really struck me is that I always just wanted to get back to research, no matter how many hats I was wearing. And in the harbor, I was wearing all the hats. I always just kind of wanted to get back to research. It really was a lot of fun, but I never wanted to give up the mentorship or the education aspect of my job. So when I decided last year to move forward with my career, I started looking into what might make me happy. And I learned about this job called a staff scientist, which I actually didn't know much about before. A staff scientist is somebody in the lab who is fully trained. You're not a student or a postdoc. You've finished your training, but so you're capable of doing all the other things, but you're not necessarily the person responsible for funding the lab, which means I could just go back to doing research and also mentoring and also educating as I pleased. And it was all the best things that I wanted. So I was very excited to kind of look for these staff scientist jobs after I learned what it was. And when I met Dr. Emily Hatch, she showed me this video, and I'll explain to you what it is after we've seen it. So if you watch the cells, these are actually nuclei of cells that you're seeing here. You can't see the cytoplasm. And they're exploding, which blew my mind, because these nuclei in these cells were exploding and surviving. And I didn't know they could do that. And here, I've taught biology for years now, and I didn't know that your nuclei could explode and survive that. And that's what the Hatch Lab works on. So before we get into the research, I want to give a little bit of background, because I understand that there's a lot of students in this talk. So whenever I'm teaching biology and I'm going through the anatomy of a cell, I'm sure you've been through this talk where we say, okay, here's the nucleus, and then around it you have your membrane and your endoplasmic reticulum. Excuse me. And when I was teaching biology, I often focused on all these things in the cytoplasm because that seemed to be where all the action was happening. The nucleus was just holding and protecting that DNA when you oversimplify it in that way. But here in the Hatch lab, we actually focus specifically in on that nucleus. So if you imagine this nucleus of this ball of goo with all these proteins and like a soupy, gooey thing, we like to focus on the chromatin, our DNA. This is what maintains our genetic integrity here. This is how we pass things down to future generations. And then there's this protein layer called the lamina, and there's a bunch of different proteins in here in this filament that goes around the chromatin. And then you add your membrane system on. So your nuclear membrane is here in gray, and it's sort of contiguous. It connects with the endoplasmic reticulum, which is where a lot of your ribosomes can be, your fatty acid synthesis can be. And so you have all this stuff on the outside, and the goal here is really to protect that chromatin and protect what gets in and out of the nucleus. And speaking of that, you have your nuclear pores, which are one of the methods by which things can get in and out of the nucleus, whether actively or passively. The pores control what's happening because you don't want to mix your cytoplasm with your nucleoplasm, the stuff on the inside. So a protein I'm going to talk about a lot today is called BAF, shown here in yellow. And BAF cross-links, so it connects to the DNA, and it also connects to this lamina. So it is responsible in part for organizing your chromatin and keeping everything where it is so that it can do the function of transcription and making your RNA, which turn into proteins, etc. So BAF is a really important cross-linker that holds your chromatin to the lamina. And I wanted to put a few more proteins up here because I'm going to say these names again later. You have lamin A, which is a part of your lamina, LemD2, which is connected to your membrane and also binds to BAF, and emerin, which is another one that binds to the membrane and also to BAF. So these we're going to refer to as Lem domain proteins or BAF binding partners. And at the end of it, to give you a nice summary, a chromatin cross-linking protein called BAF has binding partners including lamin A, LemD2, and emerin. And our goal for this project was to find out how all of those things are involved in maintaining nuclear integrity, which basically means making sure everything's okay after your cell explodes, if that should happen. So that video I showed you where you saw that quick flash coming out of the nucleus, what was actually happening is if you imagine in the beginning of the video, your nucleus looked kind of like this, and then there was a hole in your nuclear envelope, and all the stuff on the inside started to spill out. And it kept spilling out until the proteins came and they managed to repair that hole, and then everything gets reimported back into the pores, and you get this nice clean cell again. And this is something that happens, but not very often. When does it happen? It can happen during viral infection. It can happen in cancer cells. And it also can happen in things like fibroblasts or migrating cells as they squeeze through your vascular system. What are the potential consequences? So although this is something we've obviously evolved to be able to handle, what if it goes wrong or if it isn't efficient? What can happen? Well, the contents of the cytoplasm can damage your DNA. Your organelles could mislocalize. So for example, a mitochondria could wind up in the nucleus. Or in extreme cases, you could wind up with cell death. And that protein called BAF that I was talking about, that cross-links the DNA to the lamina, about a year ago, actually almost exactly a year ago, another group published that it was required for this process to occur, for this repair process to occur, because in their hands, the cells died if you took away that protein. If you took away BAF, the cells were dying. But when I joined the lab, the postdoc, Allie, had determined that that's not really what she saw all the time. She had been working on taking away BAF out of the cells so that you could see what would happen. And she was seeing a lot of survival. And so that's what we've been working on over the past year. And that's what I'm going to talk about for the rest of this talk. So how do you study something like that? It's not like it happens all the time. And usually it happens when a cell is migrating through a small pore. So you can see the cell kind of reaches out, and that's going to cause the nucleus to rupture to kind of relieve some of this pressure as the cell moves through this pore. And in the end, everything is restored and you have your cell all whole again. But it can be difficult to do this a large number of times and coax cells through tiny pores. So what we do in our lab is we actually weaken this lamina, the pink part with all the proteins around the side. We make it weaker by knocking down one of its components called lamin B1. And so we call those SHB1 cells because they've got these little holes in the lamina. And so by doing that, by using cell lines where we've knocked down lamin B1, we can see this rupture and repair process happen spontaneously. And here's what it looks like when we're looking at still images. So before the rupture, you often see these hernias that precede a rupture. In the beginning, all these nuclear contents leak out. And then towards the repair end, you see them start to be reimported into the cell, and your cell becomes whole again. So you're going to see on a lot of these slides, it's going to say RFP NLS loss or regain. And RFP NLS is just a red fluorescent protein that we've tagged with a nuclear localization signal. So it's basically representing all that goo inside the nucleus. So one way that you'll see our data is we visually determine the rupture duration by looking for when does the goo start coming out and when is it all back in. So it's all about visualizing this RFP NLS signal. Another way that we look at this is to trace the pixels from live cell imaging videos. So, excuse me, like that video you saw at the beginning, we'll actually select a section inside the nucleus and count the pixels to determine how much RFP we can measure in the nucleus. So pre-rupture, you have some amount. And when it ruptures, you have a lot less RFP in the nucleus. And eventually you start to gain it back before everything is restored. So you'll see some graphs that look like this, tracing the RFP over time as well. So one of our first pieces of data here is we actually tagged that protein BAF with GFP so we could visualize it and see what was happening. So on our top row here, you have GFP BAF. In the middle, you have RFP NLS looking at the nuclear localization signal. And then this bottom is where we've overlaid them so you can see them together. And what you can see from this top panel is that when there's a rupture, BAF localizes to that rupture during these events. And that was really important to see, that BAF is not just all over cross-linking the DNA, but if there is some sort of rupture event, it's going to localize right there, and it's a part of the process. So if you look at the bottom graphs here, these are some of the traces, and you can see that we've traced this RFP signal in red, and you see it dip down when there's a rupture, and it repairs, but even the same cell can rupture repeatedly over and over. And every time it does, you'll see the BAF signal, it gets recruited to that rupture site. And this is just another way of looking at that all on one graph. So the first thing we did was we looked at BAF and saw it go to the rupture. The next thing we wanted to do is look at what's going to happen if we take BAF away. And so what you're looking at up top is a Western blot where we're visualizing proteins separated by size. You have our control showing that we didn't mess up the loading here. And you have two different conditions, and these two panels are the same, but at a longer exposure so you can see better. The first condition is our control condition, and the second one is where we've knocked out the BAF using siRNA. And so you can see that in the control conditions, you have plenty of BAF present, but when you knock it down, there's only very little and not quite enough to do the job. So down here on your x-axis, you have which condition you're looking at, control or SI-BAF, so no BAF present. And on your y-axis, you're looking at the rupture duration. And what you can see, most importantly, is that most ruptures in control conditions are, are repaired very fast. But when you remove BAF, you get a long duration of rupture. These are taking a really, really, really long time to repair. But they are repairing, which was not necessarily expected given what was in the literature at the time. It just takes a very long time.
Could you briefly say what siRNA is for those who might not know?
Yes. So siRNA is short interfering RNA. And what it is basically a little RNA sequence that matches up with the DNA sequence that keeps it from making the protein from a certain gene. And so if you add siRNA against BAF, your cell is going to stop making BAF protein.
Awesome, thank you.
Okay. Okay, so one of our questions that came up immediately is, because we know all of this stuff is connected to the lamina and the membrane, is this really a rupture duration, how long the cell the nucleus is broken open, or is this some sort of problem with getting all the nucleoplasm and all those proteins back into the nucleus? So let's trace the longest one first so we can kind of explain this. This is an SI-BAF, which means the BAF is knocked down. And you see the cell ruptures, our RFP signal goes down over time, and it takes a really, really, really long time, about 100 minutes, before it starts to re-import into the nucleus. And you can see for all these long ruptures, the rate at which they're importing is all pretty much the same. And even when you look at control conditions, while the ruptures are so much shorter, the re-import kinetics, so how fast everything's getting back into the nucleus, that all looks about the same, which validated to us that this is definitely a problem with repair rather than re-import. So now we've looked at BAF localizing to the cells, and we've looked at the duration of these BAF-free cells and how long the ruptures are. The next thing we wanted to do is we wanted to see if we can take away BAF, but then add it back and remedy that. So we wanted to complement with wild type BAF again to see if we could rescue it.
Go ahead. There was a question in the chat. Is there a maximum time length for repair before the cell dies?
The longest I saw was several hours. I want to say I've seen around 10 hours and it still managed to get through. With the chance of survival as it goes on and on and on, often the survival rate goes down.
Thank you.
So what you're looking at here is another Western blot. We're looking at the proteins. We've got our loading controls up here. And here you see our control versus siRNA BAF. When we complemented or added back GFP, which shouldn't show anything. If we added back BAF, so it should have BAF coming back in. Or if we added this mutant kind of BAF. So again, the longer exposure is easier to see. So when we added back our GFP BAF, wild type, or mutant, you can see that they're in there. But we're still knocking down all of the endogenous or already there protein. So what is this mutant? It basically still allows BAF to bind to the DNA, but it disrupts the binding with those binding partners. So lamin A, LemD2, and emerin. It can't bind to those anymore. And as a test of this, we went through and we looked. You've got your GFP BAF on the left, and it's still recruiting to the site of rupture. Our NLS signal is still showing a rupture, and you can see the overlay here. And with our mutant version of BAF, L58R, you can see that it's still being recruited to the site of rupture. So in theory, it's still doing the same job. And by looking at this one, we can see if the ability to bind those other proteins is interfering with anything. So more quantification here. So we're looking on the x-axis, we're looking at whether this is a control condition. With BAF knocked down, did we give it back just GFP, wild type GFP, or the BAF, or the mutant BAF? And on the y-axis, we're looking at that rupture duration again. So in our control conditions, you're again seeing that short duration. And if you only add back GFP, you're not adding any BAF back, you see that really, really long duration. And so again, this is in minutes. So this, we had at least one that was over 10 hours long. It was pretty intense, this one. If you add wild type BAF back though, you see it comes back down, very short ruptures once again. So that means we were able to rescue the experiment with wild type BAF. With the mutant version, you can see that it's a lot shorter in duration than if you don't have BAF at all, but it's not quite the long duration. You don't see the long duration, but you're not seeing control conditions either. So it's kind of somewhere in between. A different way to look at that, on the x-axis here, you have the duration of the rupture, so how long it was, spinned in 3-minute intervals, and everything above 30 minutes is kind of put together. And then on the y-axis, you're looking at the proportion of ruptures. And what you can get from this is for each of those same conditions, they're colored the same. The short ruptures don't really seem to change that much. But the number or the proportion of really long ruptures is a lot higher if you have no BAF or if you just have the mutant BAF, which was really interesting to us because it suggests there's two different things happening depending on the type of rupture. So now that we saw that binding to those binding partners matters, we wanted to take a quick look at those binding partners. So what you see here is your DNA stain on the left for each of these panels. And then you have lamin A, emerin, and LemD2 stained with a fluorescent tag in the middle. And you can see the rupture signal on the right. And of course, you've got your merged version. And because sometimes you stare at these a really long time and you're like, I can't even see, there's just a bunch of dots at this point, we've quantified that over here. So this is the quantification of these image panels that you see. And for each of these, the gray bar is looking at the control conditions. The teal bar is looking at SI BAF, so we've taken away BAF. And then at the bottom on the x-axis, what you see is how much of each of these binding partners was able to be recruited to the rupture. So normally in control conditions, you get a lot of each of these proteins binding. But if you take away BAF, they're not being recruited to those sites. And so what impact would it have on the rupture duration if you took these proteins away instead of BAF? So down here, you have siRNA against each of these things listed, BAF and its binding partners, and we're looking at that same rupture duration graph again. For comparison, we've included these short control ruptures and the really long BAF ruptures. And if you take each of the binding partners away, it is a lot closer to the control conditions. But you do see, especially with LMD2, slightly larger and longer ruptures. So it does show that these binding partners are playing a role, but maybe BAF is upstream and a little more important for the efficient repair. And so looking at that same information again with the rupture duration on the x-axis here and the proportion of ruptures on the y-axis, you can see that again for all of these, no matter which siRNA you use, the short ruptures are all approximately equal. There's not much of a difference for these very short ruptures, less than 3 minutes. But as you move closer to these long 30+ minute ruptures, you see that knocking down BAF has the most long duration ruptures. And these binding partners here, where you have lamin A, emerin, and LMD2, you have more long duration ruptures than the control cells, but it's nothing compared to what you see with BAF. So what we learned in this work is that with BAF, when you have a rupture, you have really efficient repair and it goes really quickly. But without BAF, your rupture persists and there's greater leakage in a lot of cases before it's able to repair after a much longer amount of time. So the duration of rupture repair is longer when BAF is absent in a subset of repair events, because remember, they can still repair very quickly. This extended repair time isn't fully dependent on recruitment of BAF's binding partners, but they are involved in some of these repair events, as we saw that phenotype was right there in the middle. And our work indicates that BAF is that BAF influences the efficiency of repair, but suggests that there might be additional pathways. And so the last thing I'm going to show you is a video that Dr. Ali Young put together. She's the primary author on this paper, and it's going to recap everything I just said.
The nuclear envelope is a double membrane system connected with the ER. It is supported by a meshwork of lamin filaments and has many nuclear pores. A critical function of the nuclear envelope is to serve as a barrier that controls access of macromolecules to move into and out of the nucleus. Surprisingly, this membrane barrier can rupture during interphase, a time when cells are not dividing and the nuclear membrane should be intact. Cells typically repair these membrane ruptures and survive. However, these events may trigger DNA damage, inflammatory responses, and could promote cellular invasion. Nuclear ruptures can happen when healthy cells are under a lot of pressure, for example, while migrating through confined spaces that squeeze the nucleus. And these ruptures are more frequent when the nuclear lamin network is disrupted in diseases, including solid tumor cancers and laminopathies. Thus, understanding the underlying biology of nuclear membrane rupture and repair could identify new targets and treatments for these diseases. So we asked how the cell repairs ruptured nuclear membranes. One of the proteins cells use to construct the nuclear membrane is BAF. BAF binds both to DNA and nuclear envelope proteins. When we depleted BAF protein from cancer cells, we found that ruptures persisted for longer, indicating that BAF makes repairs happen faster. And we found this activity partly depends on BAF's ability to recruit other nuclear envelope proteins, some of which are attached to membrane. We think BAF is part of an assembly line that brings in membrane to patch breaks in the nuclear membrane. Interestingly, not all ruptures were equally affected by BAF loss, suggesting there could be multiple ways the nuclear membrane is repaired. To learn more, please see our paper.
Okay, and
The nuclear envelope is a
And she mentioned, please see our paper. I am pretty excited to tell you that today this morning, our paper was published in print in MVOC. So again, this was Allie's primary author paper. And again, some more acknowledgments because we've got Allie and Emily in particular with whom I worked on this. And with that, do you have any questions?
Thank you so much for that, that presentation. There are a couple questions. There was one, and this may have been answered by the video at the end, but I'm just kind of curious in your own words. If you had to just kind of distill down to the kind of strongest significance of the research of studying BAF protein and the rupture process, what would you say? Like if you had to kind of summarize the most significant aspect of that.
I would say there's two. The first is that given the context of the field when I joined the lab anyway, is that cells are able to repair. So that was actually kind of exciting. But the second part of the significance is that those ruptures are taking a lot longer to repair. And that there may be another pathway involved. No, we don't know what that other pathway is at this time.
Good, okay. How are the contents of the nucleus reimported into the nucleus when the nucleus is repaired? I was wondering about that as well.
Yeah, so mostly through those nuclear pores that are allowing things in selectively. However, I have seen in certain cancers where things aren't always recompartmentalized correctly. And so in some cases, the proteins or whatever may just be degraded and then new proteins are made to replace them. But in this case where we were looking at the nuclear localization signal, that was through the nuclear pores.
But what's the mechanism for kind of pulling things back? Like it's going through the pore, but like, how do you go out, fetch it and bring it back?
Yeah, so the nucleus localization signal actually specifically binds to those pores, and it's so a lot of proteins that are involved with transcription or nuclear processes, they have that signal, and so it's able to bind, and actually the pores can just make sure that they selectively get in.
Oh, okay. There's a question, are things ever left behind outside the nucleus?
Yeah, I believe so. That is one of the consequences if you're dealing with a lot of rupture and repair, and like the video mentioned, in some cancers or laminopathies, some disease states, this doesn't always go well. In which case the proteins or whatever would probably be degraded. And there's a cool thing in our lab that another group is looking at. If chromatin is left behind, or during mitosis or meiosis, when things are separating, if a chromatin is left behind, it'll actually form a little tiny micronucleus.
Oh, that's interesting. One of the teachers said that that was a sweet stop motion animation, and would it be possible to use that as an exemplar for his students? So it looked like maybe it was part of a lead up to announce the paper. Is that right?
Yeah, so actually the paper came out today, and that video is being submitted to molecular biology, or the video is being submitted to molecular biology of the cell, and they're going to post it. It'll be publicly available in the next week or so.
So it won't be behind a paywall though? It will be available, you think?
I don't believe so. I've seen a lot of them when I was at home, and I can't get through their payroll right now. So yeah, I can let you know when it's up.
Yeah. If we can't get through, is there another way for us to share it with students? How many cells do you generally need to rupture in order to get significant data for these experiments?
Oh my, yes, a lot. So there were days in the winter where I was looking at over 200 cells a day.
Wow.
However, every rupture event is unique, even if it's in the same cell. So a lot of times you can get a lot of information just from one cell that ruptures, 10 times. And you can analyze
Oh.
each rupture individually.
Ah. I was wondering, are there patterns after 30 minutes that you sometimes see? Like, are there any trends? Because all the data for 30 minutes is kind of together, but do you see any particular trends over 30?
Not really. So we did that blocking because the graph just got too long, going up to 600 minutes. Most of the phenotypes were the same. The only thing, I often threw out cells where the nucleus started to shrink. It was taking a really long time, and the video cuts off because, you know, we only imaged for 24 hours, and the nucleus would start to shrink. And that was really an indicator that the cell was going to die. And so that's something we watched out for. But other than that, the long ruptures were all pretty much just really long. And that was the only phenotype.
And you're using cancer cells?
Yes, we used U2S cells for these. We've also done similar experiments in HeLa cells, and there's similar experiments out there in RPE1s. The key to making them do it, though, is knocking down that lamina protein, so it makes it a lot weaker to make them rupture that often.
Yeah, I think that's related to this other question. After you make the membrane weaker, do you still need to force it through a pore to break the nucleus, or does it just break on its own?
Nope, we made it weaker so that we wouldn't have to force it through a pore. Something else we did that I didn't show, which is super cool, and again, Lyn Gunn kind of put this together upon review, is we shot some cells with a laser just to see if it would do the same thing, you know, like, was it just our system, or if we try it another way. So we did shoot lasers at cells, and that was the coolest thing I did.
I think that the Parkhurst lab does something like that too, I think, with wound healing.
Yeah, they actually helped us set it up.
Yeah. So you mentioned laminopathies. Can you tell us a little bit about, so that's disorders of the lamina, like how does that manifest itself? So
yeah,
I'd be curious to hear more about that.
The disease progeria.
Oh, yeah.
Laminopathy. So it actually, it's a lot of them are characterized by very rapid aging phenotypes in humans. Your cell, because of the weaker nucleus, you have a lot more cell death, or you have a propensity towards cancers, or so you see that a little bit.
That's really interesting. Huh. Good. I had one more question about your graphs. So sometimes you'd see like a spike up. So the x-axis is kind of the number of cells that are doing a certain amount of time, right? So you'd sometimes just see maybe one or two that would go for the long spikes. It's hard to tell how many proportionally are in your spikes.
In the histograms with the bar graphs?
No, the other ones.
The violin plots?
Yes.
Yeah. So the violin plots kind of give you an idea how wide it is there's a lot more data points that fit into that category. And when it spikes up real thin, it's because there was just like one cell at the top there, maybe a couple along the way. When we have those graphs, I wish I had my hard drive plugged in. When we have those graphs and we fill in the data points, you would usually see the bulk of things happening at the bottom, but there is like a trickle of points that would go up to the very top.
Question about if you're still involved in salmon hatchery efforts.
Oh, that was the hardest thing I've ever done in my entire life, is to leave that program. So I am not currently involved in salmon efforts. I do have all my people out there. A lot of them have wound up working in fisheries and getting jobs in fisheries, or I have two that are planning on going to graduate school soon. So so I'm still connected with the community, and I hope that someday I can be there, volunteer, and come back and do some work.
Regina is the one I mentioned to you that was involved in Citizen Salmon at Sound Bio.
Yay!
So you may have some things to catch up about related to that at some point.
Oh, it's so different from lab science, you know, and for the students that are listening, you know, in a lab, it really is, everything's so clean and tidy, and I do like that, but there's something really fun about standing at waist deep in a river and like actually netting these fish and trying to identify them in the wild. It's pretty cool.
Tim says he wants to be involved in Citizen Salmon too. So maybe able to, you know, start something up again here.
Oh, I have a question.
Regina.
So we had some students visit Cecilia Millen's fish lab, and they were like using a fine needle to inject CRISPR. I thought it was crazy that you could do that and not kill the cell. Is it a similar repair mechanism?
Yeah, I mean, in theory, yes, it's definitely a similar repair mechanism. And there are some labs that actually do poke cells, just like we do the Lamin thing, or you can put them through the cell migration or shoot them with a laser. That is one method people use.
So there are labs that are literally forcing them through tiny pores?
Yes, yes. And that's actually something that Allie, the postdoc on this paper, she's kind of playing with right now as she's building little mazes and trying to get them to go through the thing.
Do you push them or pull them?
So you add, it's through chemotaxis usually.
Oh.
So you add something the cell wants on the other side, and they kind of go through.
Cool.
Cool.
Yeah. And our teacher, Tim, is also sharing that has some experience in raising salmon and releasing them and having them come back when he was teaching fourth graders. So,
yeah, it's just the best.
You've got a couple of people in here who would love to talk to you more about the salmon stuff too, and maybe get you involved in some of the local efforts here.
Definitely. That would be awesome.
Well, we have just a few minutes left. How about some appreciations and thanks in the chat or in your reaction icon for the talk from Dr. Gunn. Thank you so much for presenting that. I learned a lot. I had no idea that the Nucleus could do that and come back from it too. That's amazing.
Right? It's so much fun.
It's so interesting. I encourage the students and teachers on this call to keep coming back for Hutch at Home. We're gonna just keep doing this. Next week, Dr. Steve Pergam, Associate Professor at our Vaccine and Infectious Disease Division and Clinical Research Division, will be talking about COVID-19. He's been part of the COVID response for Fred Hutch, working with the Seattle Cancer Care Alliance. He's really thinking a lot about prevention and treatment of infectious diseases overall. And he's also going to talk about his personal journey as a scientist and researcher. And we will have the ability to submit some questions in advance also for this talk, because I know a lot of people have questions about COVID-19. So this will be our first COVID featured talk. And so please help us spread the word about that. And then, last but not least, we have the feedback form. We hope you'll fill out the feedback form and let us know how things are going for you. Give Phage some attention now is in the chat. So Phage, so what was funny is I actually started following Dr. Gunn on Twitter before we met. And so when I met her, I was like, oh, you're the one with Phage. So I knew her from her dog. And I have yet to meet Phage in person, but hopefully someday. So just thanks everybody. We'll see you again next week. And thanks again, Amanda, for that great talk. Really appreciate it.
Thank you.